Hi, everybody. I'm Sedakut from the International Biogeography Society, and welcome to the Funk Lecture Series on Biogeography. Today we are hosting Luís Valente, and the talk title is Islands, Laboratories of Biogeography Threatened by Anthropogenic Extinction. As we're waiting for more participants, I'm going to go over a few slides, and then Sandra will introduce Luís and start the session. So the next Funk lecture will be on October 25 with Crystal McMichael. We'll announce the title as it draws near. Besides, as you all know, the upcoming IBS Biennial Conference is in Prague. So stay tuned and check out the IBS website for the latest news. And last thing is a reminder, after Luís's talk, there will be a Q&A session. And you may remember that you'll see at the bottom of the Zoom window, there are icons. You can type your comments on chat or questions on the Q&A section, or you can raise your hand to ask your question. Well, that's all for now. I think we may start, Sandra.
Okay, so it's my great pleasure having today here Luís Valente. He is an evolutionary biologist, an island biogeographer, and he's interested in biodiversity in isolated environments. He uses an array of methods from fieldwork, phylogenetics, modeling, and genomics, with the aim of reconstruct the assembly of biological communities on islands at macroevolutionary timescales. He is currently a senior researcher at Naturalis Biodiversity Center and at the University of Groningen in the Netherlands. And today is here delivering this fun lecture that is called Islands, Laboratories of Biogeography Threatened by Anthropogenic Extinctions. So, Luís, the floor is yours.
Great. Thank you, Sandra, for the presentation. Let me share my screen and remove this. This slide. The participants.
So you need to go down to the share screen.
Yep. Share.
Yes, brilliant. Fantastic. Now, yes, all for you.
Great. Let me just move this thing out of the way. Okay, so you can all see me and hear me, I hope. And yeah, thank you all for joining this Vicky Funk lecture, and thanks a lot to the International Biogeography Society for inviting me. It's a really great privilege to be giving this presentation, and thanks to the organizers of this colloquium. So in this talk, I'm going to be presenting the work that I do studying islands, with particular focuses on answering questions about biogeography and also about extinctions. So I think we all know in this audience how important islands have been for the study of biogeography in general. They've been really fundamental model systems, and they are particularly interesting for these two opposing forces that are acting on them. So on the one hand, islands are widely seen as being cradles of diversity. So they've been the setting of a lot of adaptive radiations, evolutionary innovation, so lots of kind of unusual adaptations to islands. And also they're home to many endemic species that are found nowhere else. And a really nice example of this are the honeycreepers of the Hawaiian Islands, which from a common ancestor, they have evolved into this great variety of shapes, colors, and sizes and numbers of species. However, the honeycreepers are also a very nice example for the other opposing force that is acting on islands, which is that they are hotspots of extinction. So because species have evolved in isolation on islands, islands tend to be really disproportionately affected by human-caused extinctions. And the majority of species you see on this image of honeycreepers have already gone extinct or are currently critically endangered. So this shows really well these two opposing forces. So in my research, I'm pretty interested in looking at these opposing forces, and I try to address questions about islands as cradles of diversity. And the main question that I'm interested in is, how are insular communities assembled? So how do species come together on islands? At what rates do they speciate or diversify? And we are also interested in studying islands as hotspots of extinction, looking at questions related to evolutionary history. So for example, how does the kind of thinking about the loss of evolutionary history and going beyond just measuring how many species go extinct. So, and to address the green part of this slide, you need to know about extinction, of course, because on many islands, most of the species have already gone extinct or are critically or endangered. And if you want to know about extinction and about evolutionary history, then we need to look back into rates of diversification, which are, we need to know about diversity to be able to estimate those. So, the way we address these questions in my group is we do a lot of field work. We do a lot of sequencing of island species to then build phylogenetic trees. And then we do a lot of modeling, so simulation modeling with process-based approaches. So if we go back to the example of the Hawaiian honeycreepers, they basically colonized Hawaii from the same ancestor. They've evolved a monophyletic radiation. And it can be represented by basically a phylogeny that represents a radiation on an island. And for many years, there's already been a variety of methodologies that allow us to study, for example, diversification, extinction, correlates of extinction, or ancestral state reconstructions, ancestral trait evolution, for example. However, we know that on islands, we don't just have a single colonizing lineage. We actually tend to have multiple lineages that colonized, and each of them have their own evolutionary histories. So here I'm just showing you the birds of Hawaiian Islands, and each bubble represents a different colonization event that led, some of them have radiated quite a lot, whereas others have not radiated at all, and might just be composed of just one species. So each of these can be then have their own mini phylogenies. So some of them are radiating phylogenies, whereas others are just basically a single line because there hasn't been any diversification on the island. So what we try to do is develop a method that would allow us to study islands in this context of looking at multiple phylogenetic trees from different clades. So this is what we did quite a few years ago now. We developed the DAISY model, which stands for Dynamic Assembly of Island Bioters through Speciation, Immigration, and Extinction. And this is developed with Rampal Etienne and Ali Phillimore, who are really long-term collaborators of mine and also been very important mentors for my research over the years. So what DAISY does, it brings together phylogenetics and island biogeography into a kind of a simulation model where we allow species to colonize islands at a certain rate. They can go extinct at a certain rate. They can speciate via cladogenesis, so when one species splits into two new species, and they can speciate via anagenesis. This is when you have speciation without lineage splitting. And we can then simulate this for multiple lineages. And here I'll just like to show you kind of a typical workflow of a DAISY analysis. So we start with compiling the phylogenetic data for the group that we're interested in. For example, all the birds on an island. So we compile these mini phylogenies, and from those mini phylogenies, we date them and we extract colonization and branching times, which we can then feed into the R package DAISY, which can estimate via maximum likelihood all those parameters that I mentioned in the previous slide. And then when we have those parameters, we can then simulate islands. So here you can see an example of a simulation of an island from the moment the island was born up to the present, showing how the number of species has changed through time. And this is just some brief examples of how we've applied DAISY in the past. So this here we applied DAISY to the birds of the Galapagos Islands and the islands of Macaronesia. And you can see for each that we compile those mini phylogenies, so to say, for each of the island species. Some of them have radiated, but most of them have no radiation, so they're just represented by one line. So we fit DAISY to this data. We estimate parameters, and then we can do things like this. We can simulate how the number of species changes through time on islands. And here, just as an example, you can see that the islands of Macaronesia all show a pattern of accumulation of species where there seems to be an equilibrium, kind of a asymptotic equilibrium diversity has been reached. Whereas in the Galapagos Islands, we don't see this kind of plateau being reached, suggesting that non-equilibrium dynamics are at play there. Another brief example is a study from a couple of years ago where we used DAISY to study island biogeography more on a global scale. So this time we compiled phylogenetic data for 41 different archipelagos for bird species. Then we fitted DAISY to this data, and we were able to make plots like this, where we are able to see on a global scale how area and the distance to the mainland influences all these rates of extinction, cladogenesis, anagenesis, and colonization. Recently, we've been quite active trying to test how robust DAISY is to various factors. So, because the original DAISY model does not consider quite important things, for example, geodynamics. We know that islands change a lot through time and area, and that this affects the rates at which species can colonize or diversify. And the same thing happens with traits. So DAISY is silent on traits, but of course we know that having certain traits can affect your rates of diversification, can also affect the rates at which you are able to reach an island. So in the studies with the 3 PhD students of ours, we actually did a lot of simulation work, and we were able to show that DAISY is robust to oceanic islands, so islands that were never connected to the continent, also to ancient continental islands, so places like Madagascar that have been separated from the continent for a very long time. And it's also robust to sea level fluctuations. And also, it is robust to traits in most kind of the biologically realistically scenarios that we tested. It was robust. However, it is not robust if we want to apply it to kind of continental islands that have just split recently from the continent, and also land bridge islands that are kind of connected and disconnected from the continent. And it is also not robust when we have really extreme differences in rates of speciation. So that suggests that we need to develop new models to tackle these types of scenarios. So one interesting thing is that not many people use DAISY actually, because it's quite difficult to use. The reality is it is difficult software to use. Because it uses non-standard data format. So most people working on phylogenetics, they get their tree and they feed the tree to a program, whereas DAISY can use multiple trees from different sources, needs to be formatted in certain ways, they need to be dated, we need to have reconstructions of the biogeographical history. So to make this all easier, Joshua Lambert, who's a PhD student with Jean-Paul, Etienne, and me, developed this really cool package called DAISY Prep, which what it does is it basically makes everyone's lives much easier. So if we start with a tree, this will be your kind of typical data set that people might have. So a tree with different island clades that are marked in colors. It allows you to extract these very easily to create then a data set that, as you have on the right, which is just those mini phylogenies from the island clades. So it's a series of algorithms that allow really kind of reproducible and user-friendly use of preparing the data, and allows us to incorporate uncertainty in biogeography, dealing with missing species in lots of different ways. Sometimes we don't have some of the lineages included, but we can include them, and there's always various ways. So I suggest you try this package. It's available on CRAN. So in this next part of the talk, I want to return to this idea that islands have these two opposing forces, so as cradles of diversity and as extinction hotspots. And I want to illustrate this with two case studies. So one case study for the green part and one for the red part. And as a really nice case study for as an example of islands as cradles of diversity, I have picked a really interesting group, which is the family Asteraceae. So this is the largest plant family in the world. It has around 25,000 to 33,000 species. And it's a really kind of key and famous component of many island floras. It has a lot of endemic species found nowhere else, found only on islands. It has a lot of very kind of famous radiations in plants. For example, the silverswords of Hawaii are perhaps the most famous radiation in the plant world. They also have very interesting patterns of trait evolution, for example, here. So the Asteraceae family is the daisy family or the sunflower family. And these plants here are woody daisy plants that become, so you can have in the Galapagos forests of daisy trees, which is really cool. And the list goes on and on with lots of interesting examples from different islands. I think in the context of this lecture series, it's also very important to point out that Vicky Funk spent a lot of her career working on Asteraceae, compositae. So this was her really kind of favorite group, where she was really a trailblazer, producing studies on biogeography, systematics, and evolution. And this is just an example of some of the work that she's done. So this has been really inspirational to all of us who are now working on Asteraceae. So this is an ongoing project that we're doing on Asteraceae on islands, and I want to thank all the people that are here in this image. They have helped us in various different ways with samples, with data, with the analysis. But I would like to highlight Lizzie Robel, who's my PhD student, who's doing most of this work, and also Marina, who is a lab technician has done most of the sequencing work that we are now finishing up. So we've looked at Asteraceae in lots of different angles. First of all, we looked from a diversity patterns perspective. So we obtained species lists from the GIFT database, which is a global database of island species, and added to that a few new floras that were not included in that data set. And we find that the numbers are really huge. So we have over 5,500 native Asteraceae species on islands and over 1,600 endemic species. So, and you can see in the map that they are distributed basically all over the world. But interestingly, when we compared Asteraceae to the top 10 most diverse plant families on islands, it turns out that we were surprised actually that Asteraceae is not the most diverse. It's actually Orchidaceae and Rubiaceae are more diverse on all islands. However, when we go to oceanic islands, Asteraceae then appears at the top as the most diverse plant family, which suggests that it has quite a good ability to colonize these more remote oceanic islands. We also looked at drivers of diversity from a more kind of macroecological perspective. So what is driving species richness on islands? We did a series of generalized linear mixed effects models. And here I'm showing you the results for the number of native species richness of Asteraceae on islands. You can see that area is, as usual, a really strong predictor of diversity on islands. Isolation is also a strong predictor. So the more isolated islands tend to have fewer species. And oceanic islands also tend to have fewer species than continental islands. When we look at endemism, we can see a similar pattern for area, so area being a very strong predictor. But this time, isolation and oceanic islands show the opposite pattern. So more isolated islands that have never been connected to the continent, they tend to have bigger proportions of endemic species, which kind of reinforces this idea that isolation promotes speciation. And an important thing that I'd like to say here is that both these plots kind of generally agree quite well with the classic island biogeography theory, which to me really kind of reinforces that Asteraceae might be a really nice model system for studies of island biogeography. And we also looked more from an evolutionary perspective, and we looked at the number of radiations of Asteraceae on islands. So we defined the radiation as a group of monophyletic species with 3 or more species that resulted from the same colonization of an island region. And we categorized them into confirmed radiations. So these are radiations that we can confirm that they are monophyletic using a phylogenetic tree. And then we also classify them as putative or kind of potential undiscovered radiations, which is when we have a group of species that are endemic to an island, and they are from the same genus, an Asteraceae. So potentially there could be a radiation, but unfortunately we don't yet have a phylogenetic tree to confirm that. So in total, the number of radiations is 108, and this is much bigger, for example, than the total number in birds, which is around 40 bird radiations across islands. And yeah, I'd just like to highlight here. So in black in the map, you can see the confirmed radiations, lots of confirmed radiations in Macaronesia, for example, and lots of unconfirmed radiations or putative radiations in the Caribbean islands and the Indian Ocean islands in particular, and also Melanesia. So to kind of get information on these radiations, we're doing exactly that. We're reconstructing new phylogenies for the communities of a few archipelagos. So we're focusing on the Mascarenes archipelago in the Indian Ocean, also Hawaii and French Polynesia in the Pacific Ocean. And we have done a lot of field work with lots of great collaborators on these islands. And we've, interestingly, we have to also use a lot of material from herbaria, because a lot of the island species of Asteraceae have already gone extinct, or they are extremely rare and very difficult to find. So we can only use material from herbaria. So I don't have any results to show you, but I can tell you that we have already sequenced the genomic DNA for all these taxa that we have. We used HypeSeq as a genomic technique, and we have dated these trees. And so now we are in the process of analyzing. So the next talk I give, I hope to show you some new cool results. So in the last part of this talk, I want to move on to the more depressing side of the talk, which is talking about islands as extinction hotspots. And the case study that I've chosen is a study that we've recently done on mammals of Madagascar. So we wanted to ask in this study how much evolutionary history has been lost or is currently threatened on islands, in Madagascar in particular. And this is, so we already know more or less how many species have gone extinct, or this is easier to quantify that. But what we wanted to know is how many years of evolutionary history, what amount of evolutionary history in terms of time has been lost because of human activities. And again, this has been a really collaborative study. So the study has been led by my student, Nathan, who collaborated with, we collaborated with lots of people from Madagascar, including Steve Goodman, and also collaborators in the United States and in the Netherlands. So just a bit of background. A few years ago, Jean-Paul Etienne and I and Liliana Davalos, we developed this new metric called the evolutionary return time, which basically measures how long would it take to recover the diversity that was lost from an island because of humans. So just to explain it more or less, so if you look at this image, you have the natural trajectory at which species are accumulating on an island, for example. So this is the time in millions of years, this is the number of species. And we can imagine, let's imagine that the diversity that was present on the island before humans arrived is this blue line. But then when humans arrive, they lead to extinctions. So the diversity that is present is now much lower than it was. So what DRT measures is the time that it takes to go from this red line up to the blue line. How long does it take to recover diversity back to the original diversity under assuming natural kind of rates of speciation, colonization, and extinction. And the nice thing about the ERT is that it kind of gives us a slightly different perspective. So it is different from measuring the numbers of species, and it is different from looking at phylogenetic diversity, which is kind of measuring the branch lengths of phylogenies on islands, because it takes in consideration the specific rates that are applying on a specific island, and it allows us also to compare different islands. So we, of course, picked the mammals of Madagascar. They are a really fascinating group. So Madagascar is really well known for the high number of endemic species of all taxonomic groups, and particularly in mammals. So we have groups of mammals that are found nowhere else. For example, the lemurs that everyone probably knows about. We also have the tenrecs, lots of different species of bats. And of course, unfortunately, a lot of these species are now endangered and severely threatened with extinction. So this is what Madagascar used to look like before humans. So all of these species that you see in this image have gone extinct, but we used to have giant birds. They're not mammals, obviously, but we used to have the giant elephants, elephant birds, sorry. We had pygmy hippos. We had all these species of giant lemurs, which are all now extinct. So in this study, we wanted to ask, how long will it take to go from the diversity that we have today back to the diversity that was originally there when humans arrived? And also we wanted to ask, what happens if the currently threatened species go extinct? So how long would it take to go from the diversity, if they go extinct, back to the diversity that we have today? So this should give us a measure of the different types of impact. So this is what our data looks like. We quantified how we used extant species data, but also data from the paleontological record to identify how many species were present on Madagascar just before humans arrived. And we came to this number of 249 species of mammals. And in this plot here, you can see the different colonization times of each of the colonization events. So these are, again, those mini phylogenies that I keep talking about. But in fact, here I'm not showing the whole phylogeny because they wouldn't fit. So we can see the times of colonization. You can see that in blue, it's all the bats. So the vast majority of colonizations of Madagascar were by bats. There's only 5 by non-volant mammals, including the hippos, which have gone completely extinct. And unfortunately, 30 of the species have already gone extinct. And of the species that are still alive, 128 of them are currently threatened. And before I show you the results of the ERT, I just want to show you this plot showing you how the threat status of species on Madagascar has increased just in the last 10 years. So you can see between 2010 and 2021, there's been a really dramatic increase in the numbers of threatened species, particularly for non-volant mammals. A lot of this is due to species that were not previously classified by the IUCN, but a lot of it is also real changes from a threat to a non-threat category. And here you can see the evolutionary return time estimates that we obtained for both bats and non-volant mammals. So the return to pre-human diversity with us, actually, it's quite long, around 1 to 3 million years, but it's quite short compared to the amount of evolutionary history that would be lost if the currently threatened species go extinct. So if we did this analysis back in 2010, we get around 10 million years or less than 10 million years of evolutionary history being lost. But if we go now to 2021, the numbers have risen really quite dramatically. So to conclude this part about the mammals of Madagascar, we see that around 1 to 3 million years of evolution have already been lost, and we are currently in the risk of losing 23 million years of evolution. So it would take 23 million years of evolution at least to recover the species that are currently threatened if they go extinct. And the interesting thing about Madagascar is that it has had quite low human impact so far, as can be seen by the ERT, which is relatively small for the pre-human diversity. But unfortunately, the the diversity that is currently threatened is so much larger that, of course, also the ERT is much larger. So I think Madagascar is a very good example of a place that we think, oh, it's super pristine, everything is doing great, but actually it can very, very quickly turn into a point of ecological collapse. So I think it's really an important place for conservation priority. And here, I'd just like to show off a bit the press coverage that we had for this paper. We even managed to have it on the same page as the Prince Harris memoir, which is quite funny. But the important thing about this is that it really shows that we can use biogeographical information combined with conservation to really try and raise awareness about these issues. So my last slide, just to kind of wrap up these two opposing forces on islands. So we saw that islands are cradles of diversity, and that phylogenies can provide us a lot of interesting information. I think there's a nice move away from just focusing on a single radiation on an island, but thinking more about all the different colonization events and all the small phylogenies that make up the community. And also we can use phylogenies to identify radiations. And I think, the one way to go further is to explore more and more the role of geology, ecology, and also equilibrium dynamics in this context. And about islands as extinction hotspots, we have seen that humans have had a really deep macroevolutionary impact on Madagascar. So we cannot expect that things would recover very quickly. So some people think, oh, nature is just going to return rapidly. But what we see is that, no, it's not. It's going to take millions of years for nature to go back to its kind of original state, if that's ever possible. And again, I think it's an interesting shift, again, also recently, where we are starting to focus more and more on the evolutionary history that is being lost, not just on the numbers of species. And with that, I would like to thank you all for listening. I want to thank my institutions, Naturalis and the University of Groningen, also my funding. And and thanks again to the International Biogeography Society for this nice invitation. And oh, by the way, this picture is in Bioko Island. I took it in last year, and this is an island where we're doing lots of research. And again, I'll tell about that in another talk in the future. Thank you very much.